So what's the first word that comes to your mind when you think of sharks?
Terrifying.
Yeah, so what if I told you that these deadly, terrifying creatures could actually contain a compound that could help treat Parkinson's in humans? Has anybody ever heard of the smart spoon?
No.
Yeah.
Yes.
What's the smart spoon?
It tries to control tremors when you're eating.
Yeah, exactly. It's a product designed for Parkinson's patients. And what it does is it registers the tremors in your hand and it counteracts them. So that way Parkinson's patients can do daily tasks with a little bit less difficulty. And this was designed because one of the main symptoms of Parkinson's disease is tremors. And that's included in just difficulty with muscle coordination, whether that's tremors or muscle paralysis. And another product that was designed for Parkinson's patients is weighted handles. And they're a little bit less high-tech, but it's basically just a weighted handle that's not too heavy that it makes it more difficult to do daily tasks, but rather it just kind of stabilizes and steadies your hand. So what even is Parkinson's disease? Parkinson's disease affects over 1 million people just in the US, and some of its symptoms include tremors, difficulty with muscle coordination, paralysis, amnesia, confusion, and dizziness. But a lot of Parkinson's disease is unknown. The causes of it are not entirely clear. But one of the main components that's believed to cause Parkinson's disease in humans is alpha-synuclein aggregation. And alpha-synuclein is a protein in your brain, and what it does is sometimes it can form these clumps when it's overexpressed. And it aggregates and it forms things called Lewy bodies. And these Lewy bodies are formed in neurons that produce dopamine. So these alpha-synuclein aggregations kill the neurons, and therefore less dopamine is circulated in your brain. And that lack of dopamine in your brain is most known to be the cause of Parkinson's disease, but now they're really looking at the underlying components. And that lack of dopamine is what causes most of the symptoms that are listed here. And so to address this, levodopa or L-dopa is the most effective current treatment. And what it does is it's transported to your brain and it's converted into dopamine in your brain. So in this way, it treats the symptoms, but it doesn't necessarily treat the root of the problem. And also its effects can start to wear off after a few years, but most notably, it was introduced about 50 years ago. So clearly, medicine has advanced a lot since then. And so it's important to be looking at new treatments, new things that can help treat Parkinson's disease. So based on what I talked about in the last slide, can anybody explain, one, the difference between the normal and the Parkinson's picture, and second, why they think that happened? Yeah.
There's less dopamine in Parkinson's.
Yeah, there's less dopamine. And why do you think that happened?
Because the lower the dopamine, they have more symptoms.
Yeah, well, also the alpha-synuclein aggregations cause the death of those neurons. And that death of neurons is why it's categorized as a neurodegenerative disorder. So the first paper I'm going to talk about is squalene as a broad-spectrum systemic antiviral agent with therapeutic potential, or compound found in sharks has antiviral potential. So this paper goes into why squalene can be used as an antiviral agent, but I'm just kind of using the data and the information from the paper to explain how squalene works in your brain. So what even is squalene? Squalene is a chemical that's found in the liver of dogfish sharks, and it was discovered in 1993 but synthesized in 1995, and it's important to know that it was synthesized because since it can be manufactured in a lab, it gives it more leeway to be used as a treatment. And you might be wondering why they would look at sharks in the first place. Well, sharks have a very primitive but a very effective immune system, so they thought that they might contain something that could be used as an antiviral agent in humans. So what squalene does in your brain is it has a net positive charge. So it goes into your cell and it neutralizes the negative cell membrane. And in effect, it displaces the positive proteins bound to the membrane. So by changing the charge, it kicks off these membrane-bound proteins, and a lot of membrane-bound proteins are necessary for viral replication. So that's what they kind of prove in this paper through a few different studies. But basically, just the main component of how it works on the membrane is important for what I'm going to talk about next. So are there any questions on that? The next paper I'm going to talk about is a natural product inhibits the initiation of alpha-synuclein aggregation and suppresses its toxicity. So I talked about squalene and I talked about Parkinson's, and now I'm going to talk about squalene as a potential Parkinson's treatment. So what this paper addresses is three main components. One, can squalene even affect the alpha-synucleins on the membrane? Can it have any influence on this protein-bound membrane? Because alpha-synuclein, the reason they're looking at this, they know that from the previous paper that it has an influence on proteins that are bound to the membrane. And they know that alpha-synuclein is also a protein-bound membrane, so they want to see if squalene can have a similar effect. Second, it wants to see what does it even do once it's there. Can it suppress the toxicity of those alpha-synuclein aggregations? And third, they want to see if it can even influence the phenotype. Once they prove that it affects the cell, they want to see if it can influence the symptoms that are caused by Parkinson's disease. So the first thing they wanted to see was if it can affect the alpha-synucleins that are bound to the membrane. So what they did was they tested higher and higher concentrations of squalene. So as we're going to the right, there's more and more squalene being added. And on the y-axis, it's testing the alpha-synucleins that are bound to the membrane. So as you can see, as more and more squalene is added, less and less alpha-synucleins are seen to be bound to the membrane. So this is just showing that, yes, squalene can displace alpha-synuclein from the membrane. The next thing they wanted to test is if squalene can suppress the toxicity of alpha-synuclein oligomers. So the oligomers are just the aggregations of alpha-synuclein. And so a lot of times they test the viability of cells by looking at mitochondrial damage, because like a healthy mitochondria is indicative of a viable, healthy cell. And they know that alpha-synuclein has harmful effects on the mitochondrial damage, because it is a neurodegenerative disorder. So what they tested was, well, we're mostly just going to be looking at the blue bars. So as we're going more and more to the right, there's more and more squalene added to the solution versus alpha-synuclein. So over on the furthest, it's significantly more squalene compared to the alpha-synuclein. So what they wanted to see was if more squalene can influence the cell's toxicity being done to it. So the MTT reduction, MTT is a way to test mitochondrial damage. So MTT reduction is less mitochondrial damage, therefore healthier mitochondria. And as more and more squalene is added versus alpha-synuclein, there's much healthier mitochondria. So that's just showing that squalene can effectively suppress the toxicity that alpha-synuclein has on the mitochondria and has on the cell. Are there any questions on that? The last thing they wanted to test was if squalene can influence the phenotype. So what they did was they went into the genes of a bunch of worms and they overexpressed alpha-synuclein and therefore gave them Parkinson's disease. And so the first graph is showing higher and higher concentrations of squalene again, and the motility of the worms. So they watched the worms, whether they had Parkinson's, did not have Parkinson's, and they tested how many times they bent per minute. That was the way they measured their data. And so as more and more squalene is added, the motility of the worms significantly increases to the point that the highest concentration of squalene has nearly the same motility as a worm that did not have alpha-synuclein overexpressed. So this is showing that it can really influence the phenotype. It's not just changing it on a cellular level. And The second graph is just another way of showing it, basically. They kind of mapped the movement of the worms. And as you can see, the bottom right is Parkinson's disease plus squalene. So this is the worms that had their genes changed but were also treated with squalene. That movement is much more similar to that of the worms that did not have their genes changed. So this is showing that squalene is effective in reducing the effects of alpha-synuclein aggregation and not just diminishing the alpha-synuclein aggregation. So here they prove that squalene can displace membrane-bound proteins, whether that is those helping in an antiviral, that are necessary for viral replication, or alpha-synuclein, which is causing Parkinson's disease, or a potential component of it. And in the second paper, it's proving that squalene has the potential as a Parkinson's treatment. It displaces alpha-synuclein from the membrane, it suppresses the toxicity, and it reduces paralysis. So it can affect the membrane, it can have an effect on the cell. Once it's there, it is still effective, and it can change the actual phenotype. It can change the effects and the symptoms. So this is showing that this is a new potential treatment for Parkinson's disease, which, considering the most current one was introduced decades ago, it's really important that people are looking at new ways to help treat Parkinson's disease, considering it affects so many people. And the effects of squalene are not just limited to alpha-synuclein aggregation or Parkinson's disease. It can also influence problems that are caused by membrane-bound proteins in the future. That's another thing that people can look into. And the last question it raises is, what can we learn from our environment? This compound was found in the liver of dogfish sharks, which isn't somewhere that we would necessarily look. So this questions, what else can we be learning from our environment? What other organisms around us should we be learning about and applying the ways that they react to nature to how we can fight diseases? Thank you.